Hi, I'm here to bring you guys back to the world of cultivation. My name is Sam Haiden. I work for the Agricultural Biotechnology Lab at the University of Connecticut. I'd like to thank CannMed for inviting me, and of course my PI, Dr. Gerald Berkowitz, for funding me for this wonderful adventure. So first I'd like to just go over a little bit about what we do in the lab. Our lab mostly deals with the regulation of gene expression in cannabis, and we also study gene function. What you're seeing here is some work that we've done characterizing transcription factors that induce trichome morphogenesis in cannabis. One of these is a MIB transcription factor called MIXTA, which is the focus of my master's thesis. On the top left here, you're seeing a transient expression study in Nicotiana benthamiana, where we showed the nuclear localization of this protein. On the right, you're seeing qPCR results for the expression pattern of this gene over 7 weeks of flowering. And you can see that it's peaking right about the time that cannabinoid biosynthesis tends to ramp up. And then the bottom two photos you're seeing are scanning electron micrographs of trichomes of Nicotiana tabacum leaves. And on the left, you're seeing a wild type leaf with a normal distribution of trichomes and normal branching in the trichomes, more importantly. And then on the right, you're seeing a tobacco leaf that is stably transformed and overexpressing this cannabis protein. And what you're seeing is a significantly increased number of trichomes and also increased trichome branching, which I think is the more important find there. And so basically this is an example of just what we do. We do expression in heterologous systems to identify gene function, and we use a lot of qPCR to determine how these genes are regulated, how their expression is regulated, and what their expression patterns are. We are also interested in enzymes involved in cannabinoid biosynthesis, like Lauren Sands, she's also from our lab, gave a presentation on PT4. So interesting to note too is that all of these genes are coordinated in this wonderful symphony, and that cannabinoid biosynthesis isn't actually just as simple as the biosynthetic enzymes, because those enzymes themselves are controlled by trichome genes, which are controlled by flowering genes. So there's really a complex system here, and that's more or less what we study. This here is made by Peter Apicella. He was a student in our lab, and he utilized some of my data to create a correlation matrix which shows how this MIXTA gene, it's labeled here as TFY, is tightly correlated with very important genes in cannabinoid biosynthesis, namely being geranyl pyrophosphate synthase and olivotolic acid cyclase. Many of you here probably know that those are the precursors for CBDa. And we found that the expression of this MIXTA gene was actually tightly correlated with the expression of those two genes, which to me was interesting because we've seen from other research that cannabinoids aren't really produced until the trichome produces a stalk. So I'm actually interested in seeing what trichome genes are initiating the development of the stalk, which may be also thereby activating cannabinoid biosynthesis genes. But this is just a good way to illustrate the symphony, again, of all of these different enzymes and products that are working in coordination to generate these cannabinoids that we love so much. So moving on from what we do, I'm going to get into this spectral diet question. I mentioned genetic expression, the regulation of gene expression, because how we're looking at spectral diet is a means of manipulating gene expression in order to accomplish crop steering outcomes. So we hear this buzzword a lot, achieving full genetic expression. We want to achieve the full expression of the genetic potential of the plant. People throw that term around a lot, and I don't think that they fully understand what that means, but we're going to get to the bottom of it. So the way I look at it and the way many people look at it is that your breeding and genotype selection is where you determine the roof, the potential that you can hope to live up to And the way to do that is by removing limiting factors. So limiting factors being PAR, nutrition, transpiration rates via VPD, making sure your CO2 levels are all adequate. So theoretically, by removing these limiting factors, we're allowing the plant to achieve its fullest potential. And then after you've removed all of these limiting factors, what's next? At that point, we're talking about crop steering. We're talking about actually pushing that roof and increasing the potential that we can hope to live up to by modifying the way that the plant grows with environmental signals and cues. And a lot of these cues deal with genetic regulation. So spectral diet is a method of crop steering. Arroyo defines this as being the strategic adjustment of the environment to encourage plants to grow in a specific pattern. So again, we're trying to control these growth patterns by controlling gene expression, because gene expression is at the base of so much of this. The rapidity at which a plant starts to flower is often determined by the expression levels of certain genes at certain times and how they're regulated. So one of the first examples I ever saw of this was back in my medical growing days, when my gurus back then would insist that we swap out every other HPS with a CMH. And this was really common, and it produced better flowers. Nobody really knew why, but we all presumed that it was because of the wider spectral range that you see in the metal halide spectrograph there, as opposed to HPS, which is mostly yellow. And there wasn't really data behind that. People just did it because it worked. And I think that what we're finding is there's actually maybe some data to support that. And then on the left picture here, you're seeing a greenhouse being supplementally lit by red and far red LEDs. This is a strategy that's taken up by ornamental growers, lots of growers, because red to far red ratios affect the downstream expression, the expression of genes downstream of phytochrome, which is something I'm going to start talking about in a moment. But these strategies have been taken up by other industries, not only in terms of what spectral diet to give the plant at a consistent rate, but also ways that we can give little shots of different wavelengths to elicit a different response. And this is a very complex, wide-reaching subject, DLI, photoperiod, fluence rates, etc. But what we're focused on in this study, reductive of course, is blue to red ratios and red to far red ratios. And those ratios are known to affect two signaling molecules in the plant known as phytochrome and cryptochrome. It turns out, as it happens, I think that cryptochrome may actually be the more important signaling molecule in activating flowering. But we originally were pursuing red far red ratios and phytochrome activity because it's very well studied and because it has been shown to be the origin of the shade avoidance response. If many of you have heard of that, I'm not sure, but basically the shade avoidance response is when the plant registers that it's under shaded light because of the increased ratio of far red. Far red, having a lower energy and a longer wavelength, is able to penetrate deep into the canopy, and red light is not. So the plant has evolved to know that when it gets a lot of far red, that it's in the shade. And so this phytochrome molecule becomes inactivated, and that results in what's known as the shade avoidance response, where the stem elongates and it attempts to escape that poor light to get to the good light. So knowing that that was happening, we thought that this might be a good candidate to target for manipulation to see if we can affect gene expression. Also, on the right, you're seeing a diagram of the current model for flowering initiation in Arabidopsis, which is a long day plant, and that's interesting to note because most of what we know about these signaling pathways has been studied in Arabidopsis, which has a different mechanism of flowering than short day plants or cannabis, right? And in general, short day flowering is much less well understood, much less well studied. However, rice is a very good model species that we can use to understand that because it's also a short day plant and a model species that has been heavily studied. And from that data, from that literature, we know that constans and FT are two genes downstream of both phytochrome and cryptochrome, which initiate flowering. And And those two genes, we've actually gone ahead and studied those in further studies, but basically the point is that these two genes, although they're present in short-day plants like cannabis or rice, they have a slightly different signaling methodology, a different signaling pathway, and I think that creates a lot of good opportunities for research, because in studying cannabis flowering, we can learn more about how short-day plants flower in general. So why are we doing this? Hasn't this already been done? Didn't Bruce Bugbee already cover all of this? It's a good question. So the question I have to ask myself is, why bother? What have previous studies lacked? And I think this is actually kind of glaring, like a big problem, because I've noticed that most studies done on light wavelengths are done using different fixtures. We're comparing HPS to metal halide to different LED arrays, and generally there's a lot of variables. We're not just testing one variable. These spectral diets vary in many, many different ways. For instance, this is an example of a popular study which made the conclusion that, the three graphs you see at the bottom are a study that was done which implied that the increasing blue photon fraction decreases yields. And, but if you take a look at these graphs, you'll see that from left to right, the blue photon fraction increases, but the red photon fraction actually decreases. So I'm wondering how we're able to make conclusions absolutely about blue photon fraction when there are other variables that are also changing, which have a synergistic effect. So I'm not confident in these data. I'm not confident that decreasing blue photon or increasing blue photon fraction will decrease yields, because for instance, we didn't keep the red light constant in this case. The red light decreases, and so the blue to red ratio is actually decreasing in each of these. So theoretically, if you wanted to, you could possibly say that the decreasing blue to red ratio is what's causing these yield losses. I want to shout out to Adaptive. So the way we're getting around this is that Adaptive Grow Technologies has donated these lights to us, which enable us to use the exact same fixture with different diode, with the same diode arrays, but then to either turn on or off different diodes in those lights. So we can control all of the different spectral ratios except for one. And so that's what I think, that's the advantage we have at ABL over other labs that are trying to do this. So it's ironic that I say that, because I handed this off to a student who was going to pick up the project and didn't, and I asked them to set this up to study high red ratios versus low red ratios, and they did that. Unfortunately, they did not control for the blue to red ratio, which is exactly the issue we've been talking about. So you can see here, there's a low, in T1, the top figure, there's a low red to far red ratio, meaning there's a lot of far red light, but there's also a high blue to red ratio. And I'm almost happy he did this. It was a mistake, but it ended up showing us something that's worth knowing. Because basically there's two variables, low red to far red, high blue to red in the top figure compared to the bottom. And the results of this were interesting. They were very exciting, because we actually saw a pretty significant increase in flower biomass. Leaf surface area is a phenotypic change, which is maybe not as important, but a significant and large increase in flower biomass was the result of this treatment. And so the question becomes, which of the two variables is responsible for this change? Is it the low red to far red, which is the opposite of what we expected? We thought that the low red to far red would decrease yields. Or is it the blue to red? And so that is what we need to study now, and that's what we've continued to do. So this is a new experiment where I picked this up, and I controlled for the blue to red ratio, and I manipulated only the red to far red ratios, because those are the two that we consider to be biologically significant. And the result of this was absolutely no change. Sorry for the letdown, not very exciting. But basically what we found is that red to far red ratios are having no considerable impact on yield. But that also leads us to some other interesting conclusions. So what we did also do is we studied the cannabinoids, and we found that this is fresh flower, so all that was present was the CBGA, THCA, and CBDa. We found no change in CBDa. However, by week 6, in the high red to far red treatment, we actually saw a statistically significant increase in CBGA. That's interesting because it indicates there might be some gene expression effect. But it's also interesting because we see that although there's an increase in CBGA, there's no increase in CBDa. We are concerned with what's the rate limiting step in cannabinoid biosynthesis, and this kind of gives us some information to chew on to think about that. And again, this is the same data, but this is on the composite dry flower. So instead of with the fresh flower, we just took the terminal flower. With this composite measurement, we took each plant dried and milled the whole plant together and measured that composite sample. And we still find that there's actually an even greater increase in CBGA in the composite sample. However, no change in anything else. So overall, not that exciting, but there's more. So I mentioned these genes that are flowering genes that are downstream of cryptochrome and phytochrome, and these are also very interesting targets for breeding and for potential biotechnological work because these genes are what signal the plant to start flowering. And this was interesting because, like I said, we are trying to manipulate gene expression using spectrums, not only to understand how to better cultivate the plant, but potentially to better understand how the how the plant functions and what genes are doing and how they're activating these flowering cascades. And so interestingly enough, we found that constans and FT expression were changed significantly from this light treatment. But more importantly, this is the first time that anybody's actually characterized the expression patterns of these genes in cannabis. So I'm pretty excited to say that the expression patterns of these genes so far line up quite well with the rice model. And so I feel like we could actually use the rice model to try and understand which genes are actually important for flowering in cannabis. And this is evidence to suggest that that would actually be a good comparison. Floragen is a critical night length gated gene, and you can see that before flowering at day 0, its expression was basically nothing, and then it increases to 6,000 and 12,000 fold in the first week of flower. So that indicates that these genes are probably important. However, I will say that the sad thing is that although we saw the change in expression of these genes, we saw no change in yield. So in terms of commercial value, maybe we're not there yet, but I think it's interesting to note that although these genes' expression were changed, they had no effect on yield. And so coming to the end of the talk here, this is what we're doing right now as we speak, and I think that this will be our best experiment because in the first experiment we had 2 variables in the same experiment. In the second experiment we reduced that to 1 variable. And in this experiment, we're testing the other variable. And we saw an effect in the first one, no effect in the second one. I expect we'll see some change here. In contrast to the other study that I mentioned, I believe that in this case, the increasing blue photon fraction will actually increase yields. And the reason for that is that we've kept the red constant. So what we're looking at, rather than the total fluence at any different wavelength, we're actually looking at the ratio of one wavelength to another, which I think is the more biologically significant factor to look at. So I'm really excited about this. I think it's going to turn out great. And this is, this is just a photo of that happening right now. In our grow tents, you can see on the right, you've got a much bluer sort of looking spectrum, and on the left, you've got this white-red. It looks a little bit more like what an HPS would. Because that's actually more similar to what an HPS spectrum actually looks like. So yeah, in conclusion, red to far red seemed to have no effect except for increasing CBGA, which is interesting. And although the expression of constans and FT were changed, it had no impact on yield whatsoever. However, FT and constans have very similar expression patterns in cannabis as they do in rice, so that's exciting. And also, we believe that the increasing blue to red ratio is likely to increase yields, but don't quote me on that until I get my data back, because then we'll know for sure. And yeah, big thanks to Adaptive Grow Technologies for the lights, to Gorilla Grow for donating these tents, to the greenhouse staff at UCONN, and also shout out to Austin Shep from Rapid Genomics, because he hooked me up with these gene candidates for genes downstream of Phytochrome. And thanks to CannMed. This has been awesome. Really appreciate the opportunity.